you very much for the invitation. So before I talk about what I had planned to talk, I wanted to show you this really nice data. So my lab basically studies two main subjects of interest for us are how do you detect chemicals, sense chemical, and how do you navigate chemical environment. And so we studied that both at the scale of bacteria, so where the world is at low Reynolds number, and with insect where the world has a much higher Reynolds number. And so bacteria, they tend to navigate up smooth gradient. So there's a direction of a gradient that they can try to measure and progress up in the right direction. But like flies, there's no real gradient there. What they experience is very intermittent signals or whiffs. And so it's basically, you have blank and then a strong whiff and blank and so on. And the nice thing is that with the fly, you can study very precisely what the properties of the dynamical properties of the olfactory receptor neuron that detect the signal. You can manipulate genetically the fly, etc. And so the biggest problem in this field has been how to basically measure simultaneously the behavior and the signal. So until now, it was not possible to see the signal. So the way every paper that is currently published measures the signal is that you put a photoionization detector, a device that sucks some air, and then ionize this gas, and then measure voltage difference. It tells you, okay, in this point here, I'm measuring this concentration, but when I measure it, I'm perturbating the flow because I'm sucking some air. No? And so people go and measure points, and then they say, okay, the plume is more or less here, and then they go and measure navigation. And so this
as
here,
opposed to like LiDAR?
sorry? As opposed
to like LiDAR?
LiDAR.
You can keep going up.
Yeah.
So you can measure chemicals in air in 3D volumes with this. You preserve phase, and you can do spectroscopy, but then you have timing for the photon pulses, and you can sort of map out.
Yeah.
But maybe that's not at the spatial resolution that you need.
So to do this, first we tried to use Schliering imaging, for example. You could do that, but it's very nonlinear and kind of worked, but it was difficult. We tried to use fluorescent odor, but you had to go to concentrations that were toxic. So basically, none of this worked. What works is that we discovered flies love smoke, and you can see smoke.
You can see smoke.
And we can demonstrate that this smoke is sensed by the olfactory system and nothing else. And so you can get this kind of data where for the first time you can actually see the fly and exactly see the signal that passes over. So here you get the signal, and you get the change in speed, orientation of the individual flies and its trajectory, etc. So we can now basically study the behavior conditional on the entire history of whiff, the statistics of whiff experience. And so it's a very rich data set that we work. But today I want to talk about the conflict and, if you want, synergies between phenotypic diversity, so variation between individual, and collective behavior. So, and we're going to do that with bacteria. So bacteria, if you look at an isogenic population of cells that all have exactly the same genome, if you look at the behavior, it's all slightly different. You know? And this diversity has been shown repeatedly in many different contexts to be beneficial for the population, because when you have unexpected situation, you always have one of those cells that might be able to survive. Since they all have the same genome, you basically save the genetic contact of the population, and the population can survive. On the other hand, also bacteria can navigate in group. But in order to do things in group, you have to obey some sort of rules of coordination. But phenotypic diversity, it's about not following rules. It's about being different. So individuality actually goes against basically doing things in group. So there's a tension between the two, and what we were interested in is understanding how the biological system resolve that type of tension. Okay? And so bacteria, E. coli, that we use for that, they exhibit collective migration. So for example, you can fill a tube like this with something attractive that they like, and they're going to consume it. And so in the front of a group, there's uniform concentration of that thing. They consume it, and then behind there's nothing left. And so what happens is that you get a traveling gradient and a pulse of bacteria, so a group of bacteria that moves with it. So you get this kind of nonlinear traveling wave moving down the gradient at a constant speed. Okay? But then you can take the cells in that group and put them into an environment where there's no nutrient. You wash away all the nutrients, and then you look at the behavior, and you see that individual cells are very different. The same population, you have cells that change direction very, very often, and others that rarely change direction. So I have to explain these cells, the way they move around is by doing a run and tumble navigation of a random walk. So they go straight, and then change direction abruptly, and then go straight again. And in order to move to the direction of interest, they're integrating the signal as they're moving. And when the signal of interest is increasing, they lower the probability to change direction. Okay? And so controlling this probability to change direction is controlling this probability to tumble, or the tumble bias. Function of time is how you climb the gradient. So now when there is no gradient, each individual cell has a steady state probability to change direction. And it turns out it's very different. This cell is very different than this one. You can measure these tumbles by tracking individual cells, and then if you do that for 10,000 cells, you find that the distribution of this probability to change direction is very wide in the population. There's some cells that tumble only 1% of the time, and other 50% of the time. So the question then is, how can you do this collective behavior when you have cells who have effective diffusion coefficient much larger, if you want, than this one in the same group? No? And so this is the outline of the talk. The first thing I'm going to talk about is what determines the performance of a single cell at climbing a gradient, and then how does diversity in this performance or this phenotype affect the average group performance. The second thing is then how does this collective behavior affect the amount of diversity within the group. So if you have too much diversity, it becomes hard to basically move collectively. So maybe you're going to eliminate some phenotype from the group, or there's going to be an interaction between this collective behavior and the composition of the group that can move together. No? And then the last part is going to be more theoretical, where I'd like to show you that even though random tumble navigation has been studied for, 30, 40 years, there's a kind of fundamental solution to it that hadn't been really described before, that suggests that one of the drawbacks of this random tumble navigation is that sometimes you go in the wrong direction. You're doing random walk, and sometimes you go in the wrong direction. But so there's a way to mitigate that, and that can enable individual cells to climb very fast. Okay, so the first part, how does phenotypic diversity modulate group performance? First thing we need to understand is what causes a single cell to be much faster at climbing a fixed gradient than another one. And so we've done experiments over the years showing that these differences come from differences in the level of expression of the different protein inside the cells, and then differences in this tumble bias actually are an important determinant of the speed at which the cells are able to climb the gradient. And so one experiment that we can do is take this distribution, this population of cells that has distribution of probability to change direction, and make a race with the bacteria, and then ask which cells from which beans are the one climbing faster. In this case, this experiment had done a very low dilution, so there's no collective behavior. Yeah?
So the differences are coming from expression differences within the cells, and that means that it can change over time as expression levels change, right? So what's the time scale for a green cell to turn into a yellow cell?
So right, I'll show that later. It takes like several hours, but this experiment can be basically in under 20 minutes. So what we're trying to do...
So they sort of have a relatively fixed tumble bias on the time scale of this experiment.
On the time scale of those experiments. So what we're doing is that we wash away the nutrient, and then we suspend these cells in what is called a motility buffer. This is actually a very important question. And this has been used in this field for many years. The good thing about it is that it basically gives you a snapshot of the diversity within the population and keeps it fixed for the time during which you test the performance of the individual cells. Now, if you were to do this experiment in a growing environment, which actually we're doing now, a big question is how fast the individual phenotype changes. And I have one plot where we can actually look at this. Okay, so first, like a little, why is the tumble bias basically an important determinant of the speed at which you climb the gradient? So if you think about the drift velocity of a gradient, a fixed gradient for a single cell that is basically doing runs on tumble, what matters is the average time you're going up the gradient minus the average time you're going down the gradient, divided by basically the sum of them, and the time you spend tumbling. When you're tumbling, you're not progressing. No? And so the drift velocity really depends on the contrast between the run up and down the gradient. So now if you look inside the bacterium, the structure of this signal transduction pathway has been studied for many years, it's well known. At the input, you have a sensor that adapts, and there's a negative integral feedback that controls a response regulator activity. It's called QYP. And this guy is going to control this probability to tumble of the motors. You know? And so these QYP, you can look at it as a function of time inside the cells by measuring the activity with a threat sensor. And you see that when you put a step of attractant, this QYP goes down, and so the probability to tumble goes down because the bacterium is liking what it's seeing. But then you have this adaptation. When you remove the attractant, the contrary happens and you adapt back. So this is what you see, just the adaptation feedback. On the other end of this pathway, you have this almost thresholding function. So the concentration of this QYP in this horizontal control the probability to change direction, the tumble bias or the CV bias. CV comes from the rotational direction of the flagella, which when are clockwise cause tumbles. Okay? But so if you look at any textbook or literature everywhere, what you're going to see very often is that this adaptation back to the same level is important to keep the QYP within this sensitivity region of this curve, which is a Hill function that has a field coefficient of 10. So it's a very, very narrow region, and people have been saying that, okay, you need to have this QYP in the middle there. But I was telling you that what matters is the contrast between the time you spend going up and down the gradient. So what matters is not really being in the middle of this curve, but rather this one, which is the expected duration that a cell is able to keep its direction. So that duration will, you start to swim, and how long can you swim in the same direction depend on two things. When you tumble, or rotational diffusion. Rotational diffusion will make you lose your direction. So that's why you have this plateau at some point. After about 8 seconds, those cells will lose direction because of rotational diffusion. And so what matters is, if this is the expected run length, if your average QYP level in the cell is here, when you're climbing the gradient, you're following the green trajectory. When you going the wrong direction, you're following the red trajectory, because QYP is increasing. And you see the contrast is very little. If you had a much lower, this is the tumble bias, it's much lower if you're around here. You see that the contrast now between how long you're going to run on average when you go in the right direction versus the wrong direction is bigger. No? So you want to be in the middle of this curve, which means you have to have a very low tumble bias. And so that was a surprise because everybody in the literature was saying that you should be in the middle there. But so if you then do simulation, you find that basically probability to tumble on the order of like 1% is best when you're swimming in liquid. Okay? Now we can test this prediction by doing this race of bacteria and tracking the individual cells. And what you find is basically, so the lines here are prediction from that previous model, and the dot here are average position as a function of time for those different beams. And you see the cells that have low tumble bias climb the gradient much faster than the cells that have high tumble bias. You see also that there is not a straight line, and the reason is because this microfluidics is 10 micron long. And so very rapidly you start to feel the end of the microfluidic, okay? And so that's why it slows down. But the bottom line that this shows is that in the wild-type population of bacteria, you have cells that are climbing the gradient on average 4 or 5 times faster than others. So there's wide diversity now in performance, okay? So now you could causally show that this performance can be manipulated by changing the distribution of tumble bias. So I can now modify, for example, the level of this enzyme, which is a methyltransferase that tend to activate these kinases and tend to raise the level of QIP. So I can generate a distribution of tumble bias that's similar to the wild-type population. But I can also generate one that has many more of the low tumble bias cells. And according to what we predicted before, we should see cells that go even faster. And you see that the red bean here, that is around 1% of tumble bias, is very fast. But also remember that if you don't tumble, the theory predicts that then you should not climb the gradient. So if you go lower than 1% or very low, then it should not climb the gradient. And you can do that experimentally. You see that then basically the progress is just due to diffusion. Okay? So you have a very nonlinear transition from here to basically back to just diffusion. Okay?
So tumble bias is the T given tumble.
The tumble bias
the
is
bottom,
the probability, right? Yeah, is the probability to change direction.
When you're making these model assumptions, how do you set T given plus and T given minus? Are those just known, like measured?
Oh, so the you mean this part?
The green and yeah,
This
those
here?
come from the curves that you
So
measure?
these come from the fact that, and at the end of the talk, I'm going to talk more in detail about the model, but basically you have a Poisson process in this model that control is if I tumble or run, okay? And the rate of transition depend on this QIP. And so one rate will go up in this way, the other one go up this way. And here this curve basically comes from the ratio of the two. And so your tumble bias is basically governed by the steady level of QIP that you have, which basically these two rates are balancing each other. Okay?
Well, maybe you're going to tell us as you keep going with the talk, but the question is more like, the tumble bias being low, that depends on these other variables.
Yes,
so
yes.
these just come from measurements or they come from model assumptions?
For model and measurement. I have a set of slides I didn't want to show, but I can show at the end. So we did measure in single cell the level of the protein, and then in the same cell, the trajectory, which is very difficult to do because the trajectory is over a millimeter, and then the protein, you have to use 100x. The way we did it is that we have the cells swim in a PEG deacrylate mixture with a photoinitiator. So you can track the cells under the 4x or 10x. And then you flash UV, the whole thing becomes a gel, and then you use 100x to measure basic protein content inside the cells. So we published that a few years ago. Yeah?
A question about the last data slide you were showing with comparing the non-tumbling cells to the ones, that one. So are some of the cells with high tumble bias worse than the non-tumbling cells?
They are kind of like reaching here at the very high end, basically is similar to the one that tumbled. Yeah. So then the question is now, since we have this data here, we can do something interesting. We can map the phenotype, which in our case is tumble bias, to the performance. And so this is one of those curves, and you can do that at different time points. And you see there's a very nonlinear curve. Because I was saying, when the tumble bias becomes very low, then the performance is very high suddenly. But as time passes, it becomes more linear, and this is just because of the finite world effect. And so you see those guys are so fast, they start to sense the end of the world. But now you can ask, how does diversity affect the average population performance when there is no interaction between the cells? No?
Can I ask you a question?
Yeah?
Can you explain what goes into constructing one of these gray circles? So these are individual trajectories.
So what we do is that basically we have this long channel. We have the stage of the microscope. We measure a movie, and then we move the stage and movies, and then come back again and again and again.
Yeah, but then you need to somehow
and then
stratify your data. So you record trajectories that have particular bias, let's say of 0.1.
So then each trajectory you measure basically the bias of the cells in that trajectory.
I understand. So and the error bars are so you have this whisker.
So these are standard error of the mean.
Okay. So how many trajectories do you need?
There's like about 1,000 trajectory for each dot here. Okay. So now, if you take this distribution of tumble bias, you convolve with this mapping from phenotype to performance. Because it's in a linear region here, you expect that the average performance of the group is the same as the performance of the average individual in the group. And that's what you get here. No? But if you were to do this for this guy, you would expect that the average of the diversity of the population is higher than the performance of the mean individual within that group. And you clearly see the big difference between the two. And so this is interesting because without any kind of interaction between the cell, just the fact that you have nonlinear mappings and phenotypic diversity, you can affect the first moment of a function in biology. And so this is, if you want, the first takeaway of this talk is that function is typically carried out by a group of cells, not by a single cell in biology. And you always have phenotypic diversity. And functions typically are nonlinear. And so you're always going to have this convolution between diversity in the parameter and the nonlinear mapping. And so that means that the shape of the distribution can affect the performance of the first moment. And that would suggest that the actual shape of those distributions is under selection in many cases. So just as a simple example, direct example of this effect. So now, those cells, here you see them in a macrophilic channels. They can also travel as a group if you basically provide them, for example, with aspartate inside the channel and they're consuming it and they're tracking this amino acid. And the question is, how can they move? So if you track this population of cells, you see that as a function of position and time, you see this group moving at constant speed. And then there's another group. And a lot of cells are left behind. This is done in an undefined media, Cas amino acid with glycerol. You can also do it in a defined media, which I'll show later. But how can you have this constant speed when in the same population you have all of those diversity? Okay, that's the key question. And so the question we asked was, and by the way, he was your graduate student, so he's fantastic. I really enjoyed him.
So what is the red one?
So there's two waves
So
in this
guys?
case. So no, it's just basically we want to look at who is moving in this group and who is moving in this group. Okay, so that's the question we're going to ask now. So you could say, well, maybe not everyone is traveling with this group. No? So maybe there's only a subset of this diversity that's able to take off with the first group. And so maybe the collective behavior select for a subset, and how does that happen? So in order to know that, you have to know who traveled with this group. And so we make this microphilic device where we let cells travel, enter this big area. And then when it's in there, we perfuse these, and we still with motility buffer, and then we measure the behavior of the individual cell at this point to get that tumble bias. Because here they are non-linearly responding to this gradient that I'm making, so the tumble bias will be affected by that. But here they are like in a uniform environment again. Okay? And the data is very interesting. So what you get, if you put the black lines here, and there's 3 repeats for each, because there's 3 repeats of the experiment, and you see this very reproducible. The black line is the distribution that's introduced in the device. The red one is the one that traveled with the first group, and these are PDFs, so they're normalized. Okay? So you see that the collective behavior basically selected against the high tumble bias cells, which we know are the ones that are slow in the gradient. In this case, they're all in the same gradient because they're making the gradient as they're traveling. So now you can ask the question that was asked before, like how long, maybe you were growing or changing phenotype as you travel. And so to look at that, we make sure that if you take the cells that travel that are in the red distribution and you let them grow afterwards, once they're in the big chamber, you should recuperate the black distribution. And this is what you see here. But it takes many hours, okay? So it's a selection that is non-genetic. It's really, you have a certain set of phenotype, some are eliminated, you get only those guys, then you let them grow and you recuperate the original distribution, okay? And the time scale to recuperate this is much longer than the duration of the experiment that takes about 20 minutes. So now, what is the mechanism for the diverse cells to travel together? Because there's still quite an amount of diversity in the red group. You know? And what controls the amount of diversity that can travel together? Those are like the two questions we wanted to ask. So if you think about what is happening in a very simple way, you have, if A represents like the concentration of an amino acid, and so this is the response function of the chemotaxis system, which is very well known, has been characterized. So we know the parameters of K0, K1, and M here for this chemotaxis system, and for this ligand. So F of A, if you want, is the perceived signal. And so in the front of the group, you have basically a constant concentration, and in the back, there's nothing left, and here somewhere, there's the gradient. And so now you ask, where is phenotype 1 or where is phenotype 2, etc.? And if you think about it a little bit, you find that the flux of cells of phenotype I that is moving with C, where C is the group speed, is governed by two fluxes, one that is due to chemotaxis and one is due to diffusion. The chemotaxis flux is proportional to the slope of the gradient, and this chi I, which is the chemotactic coefficient, which is the performance of a single cell in a given gradient. Huh? So some cells have a higher chi I and some cells have a lower chi I. And then you have diffusion, and diffusion is isotropic, so it's going to give you basically on both sides. So what is interesting is to ask, where's the peak position of this density profile of phenotype I? So the peak position, this is 0, and then you can divide by the concentration, and you get this rule that basically every phenotype needs to obey in order to be in the group. The rule is that its performance multiplied by the slope of the gradient at its position have to match this speed. So that means that if you have a high performer, you're going to migrate towards the front, where the gradient is shallower. If you're a low performer, you're going to be more in the back, where the gradient is steeper. And like this, basically, the cells, by spontaneously organizing themselves, you're going to match performance capabilities to the difficulty of the local task. So navigating is not the same everywhere in the group. In different positions within the group, the task is harder or easier. Another thing that's interesting is that there's an inflection point here, at which point basically the gradient becomes shallower again. So it's hard to navigate here, it gets easier, then harder again. So the inflection point will determine what is the minimum performance you need to have to be within this group. And so what limits the amount of diversity that can travel together is that at some point there's the steepest slope determining the minimum capability to travel together. Okay. So now we can test these two prediction. Do the cells organize themselves in the group? And so you can do this kind of experiment.
Were you surprised that there are only two groups?
Sorry?
How surprising is it that there are only two groups?
Only two groups?
Two groups, fastest.
No, there's a continuum.
There's a continuum.
Yeah, there's not two groups, there's a continuum. Actually, I'm going to show you now. So you can generate using different tricks. So here we're changing the level of the phosphatase, key Z, which remove the phosphate group from the response regulator. And so depending on the level of expression of the system, you get distribution of tumble bias that are like the red one or the blue one. So the prediction from what I showed before is that if you mix the red and the blue one, you're going to get the red in the front, the blue in the back, but they're going to travel together.
And this thing is on a plasmid or in a genome?
So this thing, the color here is on a plasmid, but the gene is inserted on the chromosome. And so the control of expression is on the chromosome. And this is what you get. So you get clearly that they're traveling together, but they're organized. You can now take basically the density profile of those cells, and you find that the cells that have higher tumble bias are in the back of the wave and the other one in the front. Now, if you take the green and the purple, you should get something similar, but they should be closer together, because the theory tells you that there's going to be a monotonic relationship between distance within the traveling wave and phenotypic distance. And so here you see that this is what you get. So these two are closer in phenotypic space and they're also closer spatially. So now you can take many, many different pairs like that to try to look at all of the possibilities. And you find that there's a monotonically increasing distance as a function of differences in mean tumble bias as you mix them. And you see that this organization is rather stable. So the migrating banks are basically structured. They're not well mixed within the. So now, what about the diversity, the bound on diversity? So in order to do that, what we did is that we modified the speed of the wave by basically changing the concentration of the amino acid that the cells can consume in the front, okay? If you put less, it's going to be consumed faster, and so the wave moves faster. And so by modifying going from 200 micromolar to 50 micromolar, basically you change the speed. And so and the dashed line is the prediction from the theory. And as you go to higher concentration, the speed goes down, the number of cells that can travel goes up. And the interesting plot is this one. So here we're plotting the ratio between the distribution of tumble bias that did travel divided by the distribution of tumble bias that we put into the device. No? So if you want, this is an enrichment of tumble bias. And so you see that for the slow wave, there's a kind of neutral selection. There's some selection for the low tumble bias cells and some against the high tumble bias cells. But as the waves start to go faster and faster, and also the gradient becomes shallower because it's less concentration, you start to get like a much stronger selection against the high tumble bias. And again, the dashed line here is like some prediction from this very, very simple coarse-grained model. So now, one thing that was surprising is that here we're suppressing growth as much as possible. So there's always going to be cells at the back of the wave that are falling off the wave, because you have diffusion there and then the cells fall off. And so the total number of cells within the wave will slowly decay, which means the speed of the wave should slowly decay over time if there's no growth. If you provide nutrient in addition, then they would be able to maintain constant speed for a very long time. And we're doing those experiments now. But in this case, we were not providing much nutrient. And still we were able to get like the division, the doubling time in this condition is on the order of like an hour, 10 minutes. And we're still getting like very long traveling wave. But then we realized that actually E. coli requires oxygen in order to consume aspartate. And so at the back of the wave, you have less cells. So basically, you're going to have more oxygen at the back of the wave than in the middle of the wave, because oxygen in the microfluidics is provided through the PDMS. But it might be rate limiting. And so on the side of the wave, you have higher access to oxygen than in the middle, maybe. If this was the case, you would have higher concentration at the back, so higher aspartate concentration at the back, and therefore steeper gradient at the back that helps keep the cells that are weak. And so that should enable reduce the loss of cells. And so these are the predictions from the model that with this oxygen dependency, you would get this. Then to test this, we use this dye that is wrapped into a micelle that basically responds to oxygen. And so actually that's Henry who did this work. And you see that there's a traveling wave, basically. So this is the oxygen profile that is traveling, so the different color at different time points. And then this is the density.
Curves. Can you explain what's what?
So each curve is a different time point.
Uh-huh.
And this is the density, the response of this dye. So it's giving you basically the oxygen profile that has a minimum in the center. And this is the density of cells. And so where you have the peak of the density, you have a minimum of oxygen.
So it's really tracking oxygen.
So it's basically traveling. Where you have the wave, you have a dip in oxygen, so you have less oxygen. And this is for a 100 micron deep channel, and this is for a 10 micron deep channel. Even in this case, you see that basically you have always a minimum where you have a maximum in cell density. Okay? And so this is interesting because without having any, so we also did all sorts of control by knocking out quorum sensing, oxygen sensor, because those bacteria have a receptor called AER that can bind oxygen, so you can knock it out, etc. And in all cases, basically, what you find is, so what is interesting is that without any extra secretion of an amino acid that, for example, would help cells come together or something like that, you can get this effect of boosting, reducing the loss of cells.
But it's still communication, right? Because one cell eats a lot.
Yeah. In this case, they're communicating. So the takeaway from this part of the talk is that one way to resolve this kind of conflict between diversity and collective behavior is that you can organize spatially within the group as you doing things in group. And so, and the mechanism that we found is not specific really to bacteria, because the only thing that you really need is that you have some world that you modify, and the cells that are not the first one are experiencing the cumulative effect of the cells that are in the front. And so basically, it's a very, very general mechanism.
So the
and if you
general
respond,
mechanism is global coupling through shared resource or something like this.
So the interesting thing is, if you think of the individual bacterium contribute to the signal by doing a local perturbation, but then diffusion transform that into a global gradient that basically help the whole population move. What's interesting is that the cells that are very, very fast in the front, if they were not in a group, they would not move anywhere, because you need the group to actually generate a bit like the stock market. No? If you can be very, very good at investing and you make a perturbation, then somebody noticed that, so they go and invest, and now you have a bigger signal, and suddenly you have a cascade. And the one in the front, they're going to make money because there's the cascade. Without that, not move. So this kind of cumulative effect.
Seems like you could also make an analogy with cyclists who bike together
yes,
and draft,
yes,
right?
Exactly. Yeah. And so this auxiliary cohesive mechanism is something that one should remember when using macrofluidic devices with bacteria. It can be surprising. Okay. Okay. So the last part, I want to go back to basically now a single cell. How do you climb a gradient as a single cell? How much time do I have?
20 minutes. You have 20 minutes.
Okay, so we should be able to do that. So why do you do biased random walk? You do that because you're a tiny cell, so your length scale is much smaller than the length scale of the gradient, and so you cannot basically detect the gradient locally without moving. So what you do, you basically move randomly in some direction, and you can notice if things are going up or down, and therefore you can now change the probability to change direction and climb up the gradient. The drawback of this is that you do a lot of run in the wrong direction. No? So think about just diffusion with a bias. Well, you still have your Gaussian that's diffusing in both directions as you move. So there's a lot of time lost in going the wrong direction. And this problem has been studied basically doing this kind of experiment. You attach a cell to a cover slip, you put a step in the concentration, and you measure a KYP, for example, a response to the motor. But and you get this kind of data, this classic data from Howard Berg. So you have the probability to change direction, you put a step of attractor and it goes down, and then it comes back up. You know? This is your tumble bias, this is how much you amplify the signal, and this is how long it takes you to adapt. So now, in reality, when the cell is swimming around, this is what's happening. You know? So when you do a tumble and pick a new direction, you're changing the input. You're going to change what you're going to experience next. So what you experience depends on the past decision. So there's a feedback of the behavior decision onto the signal. And this turns out is actually really important to this problem. And so there's been a lot of studies of bacterial chemotaxis going from the single cell to basically deriving a continuous equation. There's like hundreds of papers on that. No? And most of them, practically all of them, they use at some point mean field approximation, which assumes that the internal state of the bacterium is not too far from basically the peak of the distribution, so that the distribution of internal state is well behaved. Okay? But when you do this, imagine I'm climbing up the gradient, so I lower the probability to change direction. So now, on average, I'm going to keep on going up the gradient, which means we'll further lower the probability to change direction, which means, so there's this positive feedback that can drive the internal state down to zero. Basically, QIP can drop all the way to zero. So this positive feedback can drive enormous fluctuation in the internal state, breaking this mean field approximation. Okay? So we were interested in studying that and see, okay, is there some interesting effect or that comes from there? And so the little minimal model we're going to use is the following one. So we're going to assume there's an internal state F that relaxes to a basal level F0, so that's your adapted level if you have no signal, with a timescale Tm. And then it has a sensor, and so you're taking a Lagrangian derivative of the perceived signal, and this phi is what I had before on the slide, is this log of the concentration that the cell is experiencing. And n is how much the receptor are amplifying the signal. And so now this internal state is controlling this probability to switch from run to tumble or tumble to run. Okay, so those are Poisson processes governed by this F. And so the probability to run, on average, is going to be the ratio between the rate to tumble divided by the sum of the two rate, and you can write this like this, where h, if you want, is the gain of the motors. So remember, there's this very sharp curve at the motor output. No? And so the other thing that we're assuming is that when the cells are running, they're basically experiencing rotational diffusion. And when they're tumbling, they're experiencing much greater rotational diffusion. So basically, the rotational diffusion coefficient changes the problem. Okay. So now, to make the system to make the calculation tractable analytically, we're going to assume that those cells which are log sensing are in an exponential gradient, so that they have the gradient has a length scale L. Oops, sorry, it's an exponent L. And so this whole term here becomes basically S divided by L times N. V naught is the speed when you're swimming, and S is the cosine of your direction relative to the direction of the gradient. Okay? And we're gonna normalize basically the internal state by this stage. And so there's four key timescales of interest in this problem. One is the direction correlation time. So how long does it take you to lose your direction? And you lose your direction because of rotational diffusion and tumble. And so it's going to be n here is the dimensional of space. So in 3D, n minus 1 is 2. R is the probability to run. dr is the rotational diffusion when you're running. And that's probability to tumble. And that's the rotational diffusion when you're tumbling. The other important timescale is the timescale of the response to the environment. So if you swim faster, you're going to experience a stronger increase in signal than if you swim slower, or if your gain is higher, or if the length scale is shorter, the time scale becomes smaller, okay, and vice versa. Then there is your memory time, or the relaxation time of the internal state. And then there is the switching time, the run and tumble, that is on a much shorter time scale. So you do run, tumble, and it's this one. And so we're interested in the regime where what happened over multiple runs and tumble. So in the case where basically this is much shorter than these other three time scale. And so because there's three time scale, we can define two non-dimensional parameters. So we're going to use as unit of time the memory durations, the relaxation time of the internal state. And then tau E is going to be the time scale of the response to the environment in these units. And this one is going to be tau D, is going to be the decorrelation time. Okay? And so you can, if you can rewrite this problem, if you take a derivative of this, you can show that you get an equation like this where basically the change in the probability to run has now a term R times R. Here you have this positive feedback. Okay? And there's two terms, a negative feedback which has to do with the adaptation. So adaptation is trying to bring f back down to f0, but the gradient is bringing you away from that f0.
Can you remind again what is the definition of R?
The what?
The definition of R.
Of R?
Yeah.
Of R is basically the ratio between the probability to tumble per unit time divided by the sum of the two.
Can you explain again the nature of the positive?
Say it again.
What is the nature of the positive feedback?
So the nature of the positive feedback is that when you start to run in the right direction, you lower the probability to change direction. Since you don't change your probability to change direction, you keep on going. So you run longer in the same direction. And therefore you see a further increase in the signal. So this further increase in the signal will cause further decrease in your probability.
So I must have been not reading the papers for a while, because what I remember from Berg is that when you move in the right direction, you do not lower the probability of tumble, you do not change things.
No. So what he says is when you go in the right direction, you lower the probability to change direction. But he says when you go in the wrong direction, you just go back to baseline.
Yeah.
I'm going to show that I'm not sure that is totally correct.
Okay.
It's going to be one of the consequences of what I'm saying, depending on the regime in which the cells are. But so here you have the negative feedback. So you have an adaptive system, and then you have this positive feedback of your behavior that's pushing you away from your adapted internal state.
And what's the
And if
shape
you
of
take the
FOR?
ratio, sorry?
What's the shape of FOR?
Of f of r, the shape of f of r is f here is let's see, shape of f of r. F here is an internal variable. Okay, so the relationship between f and r is basically looks like a Boltzmann distribution. But I didn't put it on the equation. So the important thing is that you can take if you take the ratio between those two things, you find that the strength of this ratio is actually that parameter that we identified before. There's these two parameters in this problem. And so this tau E is basically the strength of the negative feedback divided by the strength of the positive feedback. So now, if you look at the literature, every paper that has been written in the limit where the negative feedback is dominating. If the negative dominates, then basically the internal state is always around its F0, no? Or close to its mean, basically. And so what happens when tau is is smaller than 1? That's the question. And so you can write a Fokker-Planck equation for this problem. So you have the probability to have internal state F, direction S, cosine of the direction, position X, and time tau. And you're going to have three flux in probability. One is the internal flux in F. One is a random walk on a sphere because of rotational diffusion and so on. And the other one is just the flux up the gradient due to your motion on average. No? And you can solve actually, because you're in an exponential gradient, the system reaches steady state. And so you can solve this at steady state, and you can actually solve semi-analytically this problem. We can get analytical solution. We can also do numerical simulation of it. And so if you simulate this problem for the entire space of tau E and tau dt, you find that the speed of the cells become very, very large in the region where the positive feedback is dominating. Okay, so most of the results that exist are in this region where the negative feedback dominates, and the progress up the gradient is on the order of a few % of the maximum speed of the cell. So Vd here is in units of V naught, of the speed of the cell. But you see there is a big peak here. And so first of all, in this axis, you see that the peak is around like order 1. That makes sense because this is when your memory duration is on the same time scale as the time scale of losing your direction. And that basically was expected already from previous work. But you see this big boost. Now it's interesting to look at stochastic simulation, agent-based simulation of this problem at different points of this diagram. So if you look here, you see that the cells are actually climbing like ratchet. They go a long time and then a little bit, but they do a lot in the wrong direction. And then they resume to go up. But like when you're here, you're basically doing bias diffusion. Okay? Even more interesting is to look at what's now the internal state distribution. The internal state in this region, as expected, is well behaved, and therefore mean field theory should work, and it works. But in this case, you get these very long tails in the internal state distribution. So you have large dynamic inside the navigator. Okay? So now, can we understand?
Does it ever become pronounced by model?
Sorry?
Does it ever become?
Not really, no.
Yeah, that's why I asked you about the shape.
So now the next thing to understand better what happened, we can switch from a Fokker-Planck representation to a Langevin type of representation. So where the variable has this probability to run, v is the projection of the speed on the correct direction, so it's v naught times the cosine of the direction. And this term here, which is the noise, is coming from tumble and rotational diffusion, and this is the deterministic part. So now if you just study the deterministic part of this system, you find that the relaxation matrix is like this, and this is really interesting. So you see that, first of all, the diagonal is negative, so you have, and so in that fixed point, basically the system is stable, but it's this off-diagonal term that has this tau e again. Now when the negative feedback dominates, tau e is very large. So this thing becomes 0, and so now you have a diagonal matrix, you have exactly normal orthogonal eigenvectors, and this is the space of the dynamic. So in R, that's the probability to run. This is the speed. Where you want to be is in this corner. No? You want to be moving the right direction and continuously running. The color is the strength of this noise. And you see that basically your fixed point is here, and you always tend to come back, but you're pushed away from the noise. And if you look at what the system is doing, it's basically going up and down, and a little bit, on average, a little bit up, but that's it. What's very interesting is what happened when tau e becomes small. Now you start to have circulation in this space, because now your eigenvectors are not normal, so you can have non-normal dynamics. Even though the system is still stable, you can have transients that are large. Okay. And the second thing that is cool, so you have non-normal dynamics, and you have this quadratic term here in the speed that breaks symmetry. So when you go in the right direction, v square increases your speed. If v is negative, v square will basically bring your speed to 0 very rapidly. And so the combination of non-normal dynamics and this nonlinear amplification enable the system to basically go and hang out much more up in this corner than in this case. And so the prediction there is that basically the cells should be, when they are in that regime, this long memory type of regime, they start to basically ride climb the gradient like a ratchet. So now you can look at the experiment. So this is, again, the same type of experiment in this gradient. I just put like 10,000 trajectories, but you can look at individual cells, and many of them have this climbing, and then they stay, and they keep on going, but they don't go very much in the wrong direction.
Now, these
Of
are
course,
real-
I selected.
These are real observations. not
Those are real
simulations.
cells, yeah.
Okay.
Those are real cells. And then, so we said, okay, we had all of these experiments before in this gradient, no? And so we looked at the bottom of the gradient, since they're log sensing, so at the bottom of the gradient is actually a very steep gradient. And in the upper part of the gradient, it becomes shallower, no? And so at the bottom of the gradient, the cells are basically in this region, and then they move more into that region. And so you see that definitely there's going to be some cells that are able to basically use or experience this kind of like enhanced rapid climbing of the gradient. Okay? So the summary here and the take-home message is that the classical drawback here is the wasteful run in the wrong direction, but you can exploit the nonlinear dynamics of this actually run on tumble navigation is the simplest possible one that we took. We just have a negative integral feedback, rotational diffusion, and constant speed, and tumbles. That's it. No, there's nothing else in this problem. And the key parameter is basically the ratio between the negative feedback and the positive feedback. And so we're very interested in looking also in the fly system, if something is, because we think that in terms of navigation, you're always going to have this, no? Because if you navigate somewhere, you make decision of how to move. If you make the right decision, the stream of information you get is getting better. So that will feed back onto your decision-making apparatus. And so we want to see if there's some sort of general principle behind this. And with that, I'd like to thank the people who did the work. So I had, let's see here, Henry, who is this person, who did basically the work on the wave. Then Jun Jaja did the last part, and he's basically working in Wall Street. Left side, unfortunately for us. And then Nick Frankel, Adam Waite, and where is he? Yeah, here. Jan Dufour did the experiment with the straight gradient. Then Xiongfei Fu and Setsu Kato did the traveling wave into